Okay, any questions? The recording, do the screen share. Okay, that I need to get rid of. Let's go ahead and go to here. So we're going to be working a little bit ahead of schedule. I'm going to start chapter 7. We'll try and go through chapter 7 slowly. We will spend all of, boy, that's all of this week. So we'll spend the rest of the week on this. All right. Chapter 7, the control of the microbial growth. This is actually a chapter that students have an easy time with because you guys know a little bit about disinfectants and antiseptics. And this chapter is a little bit more than that, but that's the main part of the chapter. And students just have an easier time understanding chapter 7 than, say, antibiotics. And I've just noticed that over the years that this seems to be an easy or easier chapter for students. So, as usual, I have my major goals and a rough outline. Know the terms. We've already talked about these terms in the lab, so you should know what sterilization means, what disinfection means, what antisepsis means, what degerming is, and I think that might be a new term, sanitization, autoclaving, pasteurization, and then biocide and germicide. Any questions about any of those terms? We will cover them, but you should know the terms. Be able to discuss the factors that influence microbial death rates. So if we're using an agent on a microbe, discuss the factors that influence the death rates. Be able to discuss the 3 main ways in which antimicrobial agents kill or inhibit cells. Understand the various types of disinfectants and how they work. And understand the relative resistance of the major microbial groups to antimicrobial agents. Any questions about any of that? Blow this up a little bit. Yeah, but it's not going to work. I'll go to the screen here. Let me see if there's anything else I need to look at. Nope, we're good. So when we apply a disinfectant to a population of microbes, there's a constant logarithmic rate of the cells dying. And if you plot it on a log scale on the y-axis and then a arithmetic scale on the x-axis, you'll see a constant rate. So with 1 unit of time, you get 1 log decrease in killing, or 90% of the cells dying. And with another unit of time, you get another 90% killing rate. So in another sense, if you only have 10% of the cells surviving after 1 minute, and then after another minute, you only have another 1/10 of the cells surviving, or 1/100th of the cells surviving. And microbiologists like the log plot, semi-log plot, because the x is actually arithmetic, because this is an easy equation to solve. It's y equals mx plus b. And you could use a direct arithmetic plate, but then it's not a nice curve and sort of exponential. So the longer you expose a population to the disinfectant, the more killing you'll get. And it's an easy mathematical equation. So if you have 10 to the 6 cells and you get a constant logarithmic die-off rate, meaning only 10% survive with each unit, then you know to kill off all of the cells, 1 minute wouldn't be long enough, 2 minutes wouldn't be long enough, but if you were to go 6 minutes, you would kill off all but perhaps one cell of 10 to the 6 cells. Any question about that? Okay? So if you know your starting population and you have a graph something like this, then you know that the cells will die at about this rate. Any questions about any of that? So if you know your starting population and you know that there's a constant death rate, it kind of tells you how long to expose the population to kill off all the cells. And this is something that's very useful. Like if you're in the food prepper industry and you want to kill off all the cells, of course they won't have 10 to the 6 cells, but it tells you how long to expose it. And when we're coating our lab benches, actually you don't go into the lab to do this, but if you were to do that, on the lab bench there would be maybe 10 cells or at most 100 cells, and it tells you that really all you need is about 2 minutes. Is that right? Yeah, that's right. About 2 minutes is all you need to kill off everything on the top of your lab bench. Any question about that? The rate of the microbial death is similar in that you always have the die-off happening something like that. And when you are looking at the variables to determine what cells are living, it'll be based on the number of microbes start at the present. So if there are higher microbes starting at the present, you'll need a longer time to kill off all the cells than if there's a lower number of cells starting at the present. The environmental influences can affect the microbial death rate. So most agents are inhibited by the presence of organic matter. And if ever you're working with a clinical specimen, you'll have organic matter around. That could be like dandruff on a patient. If you are cleaning up a fecal spill, well, the feces or the organic matter, and that can inactivate the agent that you're cleaning with. Like a common disinfectant is bleach, and bleach is inactivated by organic matter. And so you need more bleach if there's more organic matter present. Any question about any of that? The temperature will affect the killing rate. Generally, the higher the temperature, the more killing you'll get. The presence of biofilms will affect the kill rate. If there's a biofilm present, then you'll need a much longer time, much longer exposure time to kill off all the cells. Biofilms typically protect the cells underneath the biofilm, so if they're deep down in the biofilm, those cells will be protected, as opposed to the cells which are on the outside of the biofilm. They die usually when they're being treated, but the biofilms are really difficult to kill all the cells in them, and that can cause a problem when we're treating them. The time of exposure obviously will depend on how many cells are present and what the cells are. If you have cells that have endospores, they will be much more difficult to get effective killing. And it may be ridiculous the time you need to expose it. Like if you're treating to, let's say, carboxylic acid, for example, or some phenolic compound, and you're using it on an agent which has endospores, the time to kill all of those endospores may be centuries, okay? And so there would be no way you could kill off all of the cells. Now it depends on the agent you're applying and the endospores to how resistant they will be, but generally endospores are resistant to many disinfectants. That's not true for all disinfectants, but for many. Even bleach, which is highly effective on killing most cells, bleach does not kill some endospores. It depends on what endospores we're talking about, but so if you have certain endospores, they may be resistant to bleach, and you'd have to expose them to like centuries of bleach before you'd kill all the cells, and that wouldn't be possible. But there are other endospores which are sensitive to bleach. You just need longer exposure time when there's those endospores than if there's just regular cells. And like I said, not all disinfectants are, what's somebody saying? There are certain disinfectants which can kill endospores, and mostly that's the toxic oxygen molecules like, what's the name? Hydrogen peroxide and stuff like that. Those are effective at killing endospores. And we'll talk a little bit more about that when we're talking about endospores. So there are some disinfectants that do kill endospores, but there's very few of them. And like I said, they're mostly the agents that have toxic oxygen molecules that can kill the endospores. So the microbial characteristics can have effects on the microbial death rate, and the biggest one is, like I said, endospores. If the cells produce endospores, it'll be difficult to kill all the cells. You may not get killing within a reasonable time frame, and another characteristic, like it would be mycobacterium, the mycolic acid in the cell wall can affect the disinfectant's kill rate. If the disinfectant is water soluble, it's not going to get through the mycolic acid of mycobacterium, and so you may not get effective killing. Any question about any of that? We'll talk about the last one, the microbial characteristics, later in the lesson. So you should know the terms for the control of microbial growth: sterilization, the destruction or removal of all forms of microbial life, including endospores. This generally, when we use the word sterilize, we generally do not mean prions. Prions are really difficult to kill, and that's a topic I never got into, but generally when we say the media is sterilized, it does not mean that the prions have been destroyed. Any question about that? Prions are really, really hard to kill. You have to apply like 1,000 degrees Fahrenheit to kill prions, and it's not well understood why prions are so difficult to kill. Because 1,000 degrees Fahrenheit will incinerate any protein. So why the heck are prions so difficult to destroy? And that has never been answered. Nobody has a good answer about that. So when we're talking about sterilization in the lab, how that's done is usually autoclaving, steam under pressure. And if you don't know what an autoclave is, it's just a giant pressure cooker. But it can be done with other agents if they're susceptible to those agents, like ethylene oxide can be used to sterilize, like expensive, although it would have to be a surgical instrument that could not take autoclaving, and I can't think of anything at the moment, but some like tubing that can't be put under high temperature, you could sterilize it with ethylene oxide. Commercial sterilization is just sufficient heat to kill most agents that are coming into that can cause like food contamination in the food industry, and that would be sufficient heat to kill endospores like Clostridium botulinum in canned foods. And we won't run into that one really, because we're not going to talk about that. Sanitization is something similar, and that's just treatment of lower microbial counts where you're not really killing everything, but you're reducing them to safe levels. And that would be an eating and drinking establishment, which that's like putting utensils in the dishwasher and then serving them to your customers, the plates and the silverware. That's sanitization. It doesn't really sterilize everything, but it lowers the microbes to safe levels so that they can be used in the food industry. Any question about that? So it's similar to that, but different. Disinfection, destruction of vegetative pathogen. Disinfection generally does not kill endospores, and we're implying a use of a disinfectant when we do disinfection. So this is treatment of something that's inanimate, not a living surface. When we're treating a living surface, it's called antisepsis, destruction of vegetative pathogens on living tissue. Generally, that does not kill endospores. Any question about that? So, with necrotizing fasciitis, I don't know if this is still the preferred treatment or not, but when somebody had it on their skin, at least in the early days, and this is going back to way back to 1980 and 1990, they would actually wrap a rag and soaked with bleach around the patient who had necrotizing fasciitis. So that was one of the treatments for treating necrotizing fasciitis, and I don't know if they still do that or not. But my point is that was antisepsis with bleach. Any questions about that? But then again, when we're using bleach to kill or clean or disinfect a toilet, it's called disinfection. So you can have the same chemical causing disinfection of an object or antisepsis of your arm with necrotizing fasciitis with bleach. Any question about that? Usually they are different chemicals. The disinfectant is more harsh and it's used on objects, and antisepsis, it's a less harsh chemical and it doesn't cause damage to the patient or the skin. Okay? So like we don't usually clean the toilet with iodine, but we do clean living skin with iodine. Any question about that? Degerming does not kill all or sterilize all living objects, does only reduces the number of microbes in a limited area usually. like when you're swabbing somebody's skin with alcohol and then giving an injection in that swab site. That's degerming. It's reducing the microbes in the area to make the injection site, but it does not remove all microbes from the area. Any question about that? All right. So when we're using a chemical for antisepsis, we call it an antiseptic. When we're using a chemical for disinfection, we call it a disinfectant. Biocides or germicides are agents that kill living microbes that may not kill everything and usually does not kill endospores. There are some exceptions. Like I said, hydrogen peroxide would kill endospores. Okay, but most biocides and germicides do not kill endospores. A bactericide is an agent that kills bacteria. A fungicide, an agent that kills fungi. A viricide, it inactivates viruses. Viruses, we're not going to talk about whether they're alive or not, so that's why we're saying it inactivates viruses rather than kills viruses. Bacteriostasis, that's inhibiting the growth of a microbe, but not necessarily killing those microbes. And we talked about that with some antibiotics. Some of them are biocides, they kill the cells, and some of them act as bacteriostatic antibiotic agents, meaning they inhibit the growth of the microbe but do not kill the cells. And in the zone of inhibition, you cannot tell that from the zone of inhibition. There you have to use some other way to determine is it a biocide or a bacteriostatic agent. Well, that applies to the antimicrobial agents also. Some of them are biocide and some of them are bacteriostatic. Once you remove the bacteriostatic agent, the growth may occur. I cannot for the life of me think of a disinfectant which is bacteriostatic. Does anyone know of an agent where we could have something like that? Well, here's one that would work, and that is...
How about like a triple antibiotic ointment?
Yeah, but what ointment? It's
Like
bacteriostatic.
Neosporin.
What?
Neosporin, maybe?
Neosporin. I don't know if that's biocide or bacteriostatic. It has antibiotics in it, and I don't know how those act. But I can think of one now that I think about it, and this is actually a experimental treatment for a patient who has MRSA or MRSA, multi-drug resistant Staphylococcus aureus. And if it's resistant to all known antibiotics, you can apply honey to the MRSA infection. And that's bacteriostatic, preventing the MRSA from growing. And it's actually fairly effective. It stops the growth and it allows the host immune system to then go in and kill off the cells. Now the trouble is, once you stop applying the honey, the bacteria, the MRSA, can grow. And I don't know how they apply it. They must apply the skin and then wrap it up so the honey is staying on the MRSA infected region. I don't know how that's done, but it must be long-term exposure. And then the host immune system has time to remove the MRSA. And then generally the patient gets better. Another bacteriostatic agent would be if you put a high salt solution on the bacteria. For some bacteria, they will not die under a high salt solution, but that prevents them from growing, so it's bacteriostatic. Okay? And then when you remove that treatment, the bacteria can continue growing. Any question about any of that? Sepsis refers to microbial contamination, so you want to have asepsis whenever you're doing surgery. The absence of contamination by unwanted organism, like on the scalpel, you're going to do surgery. You want to do that under aseptic conditions. And aseptic techniques are important in surgery to minimize the contamination from contaminating microbes. Any question about that? So when we do surgery, we usually will pass the air through a filter so that the air will be sterilized, and the doctors and health professionals will be wrapped up so that obviously their skin will have microbes on it, but their skin shouldn't touch the patient. And with the mask on, they shouldn't breathe out contaminants. And then the instrument will be sterilized before use. Usually the instrument will be autoclaved and then will be opened right before surgery. Okay, any question about any of that? Different microbial agents can have different actions. Antimicrobial agents tend to kill cells or inhibit the growth of cells by doing three things. They can alter membrane permeability. You can damage the plasma membrane, either the lipids or the protein, and then cause the cellular contents to escape, and that'll kill the cell when the cytoplasm leaks out of the cell. Any question about that? You might damage the membrane and then interfere with normal metabolism. I can't think of an agent that does that. Alcohol would be the closest thing I can think of, but that tends to do more than just damage the membrane. But it would damage the membrane and then interfere with normal metabolism of the membrane. The thing is, the alcohol will go into the cell and will damage proteins in the cell. You can also damage intercellular proteins, and this would be a better example of how alcohol works. It denatures the proteins, especially the enzymes. You can target disulfide bridges in an enzyme, and certain antibiotics do that. Or you can damage the protein just by breaking the hydrogen bonds in the enzymes of the proteins, and then the protein won't have the correct shape and will no longer act. And alcohol would do all of these, okay? Damage the protein, so the protein is inactive. What's the term I'm looking for? Denatured. You can damage the hydrogen bonds in the protein, so the enzyme's in the wrong shape and it won't work. And then you can also damage the nucleic acid. That often results in cell mutation, resulting in cell death. And that's how radiation damages cells. UV light, X-rays. I'm not thinking of another agent that damages nucleic acid, but that would be one way. Actually, oh, what is that? There are some chemicals which are mutagens that can damage a cell by damaging the nucleic acid. But generally, we don't want to be exposed to those chemicals. Benzene would be an example. Any questions about that? So microbial agents tend to work in one of three ways: damaging the membrane permeability, damaging intracellular proteins and damaging nucleic acid. And different agents will act differently. Polymyxin B only puts a hole in the cell membrane, and that's how the antibiotic works. It doesn't do other things. And as far as I know, radiation only damages nucleic acids. There are different physical methods for, don't know why I'm continuously losing that. There's different physical methods for controlling microbial growth. You can apply heat, you can apply filtration, low temperature, and that stops things from growing but does not necessarily kill cells. Like if you were to put bacteria in the freezer, about 50% of the cells would, upon thawing, be viable. You can use high pressure to kill bacteria. You can use desiccation, osmotic pressure, and then radiation. So all of these are physical methods we have for controlling microbial growth. They would not necessarily kill all cells. Like osmotic pressure would not kill endospores. Desiccation would not kill endospores. I don't know about pressure. I just don't know. A high pressure and high temperature will kill endospores, but I don't know about high pressure. Low temperature would not kill endospores. Filtration would separate out the endospores. They would be caught by the filter. And if you apply high enough heat, you would eventually kill the endospore. But you'd need a high temperature heat to kill all endospores. Any question about that? Radiation would kill endospores, but endospores would take a longer exposure to radiation to kill the cell than a bacteria cell, a normal vegetative bacteria cell. Any question about any of that? So heat, all methods of heating cells do kill microorganisms, and they do that by denaturing the proteins and the enzymes. Though high enough temperatures of heat, like when you're flaming something or incineration, it actually turns the cells into char. So you're doing more than just denaturing the proteins. There's two types of heat, wet heat and dry heat. Generally speaking, wet heat is more effective, like boiling is an example of wet heat, and autoclaving and steaming. Dry heat, an example of that would be baking, although extreme dry heat would be flaming, where you put something in a flame or you incinerate something. And so this is at high temperatures. Okay. Any question about that? Most heat denatures or coagulates proteins and enzymes. An example of coagulation is when you're cooking an egg and you coagulate the egg whites. A boiling liquid like 100 degrees Celsius or 212 degrees Fahrenheit, or flowing steam, kills vegetative bacterial pathogens, most viruses, fungi, fungal spores, and you only need about 10 minutes at this temperature, 100 degrees or flowing steam. Endospores will survive that temperature and that treatment. Bacteria cells, most of them will be dead by 10 minutes, and they're often much faster. Like you expose E. coli to 100 degrees Celsius, and in a minute or two, almost everything will be dead. You don't need 10 minutes. But 10 minutes is just to kill most cells and a large number of cells. But endospores would not be killed by that. Some viruses may take longer to destroy, and like I said, endospores, they would survive greater than 20 hours in boiling water. So generally, boiling would not kill endospores. Any question about that? So reliable sterilization, you need moist heat at a high temperature and a high pressure, and that we call an autoclave. It's like a giant pressure cooker, steam under pressure, very effective. You're applying 15 pounds per square inch of pressure. I don't know if you understand that or not, but it's just a lot of pressure. Applying 15 pounds per square inch at 121 degrees Celsius. That kills all organisms in about 15 minutes, including endospores. So 1 liter of solution at 15 pounds per square inch at 121 will kill all endospores. Now, if it's greater than 1 liter, you might need more time or higher pressure. Steam is very good at sterilizing the surface of something, but it only sterilizes the surface. Like if you had something that's in 3 dimensions, and you can sterilize the surface of the object, but not necessarily inside, because the steam is not touching inside. Steam is usually hotter than 100 degrees Celsius, and exactly what temperature it is, but it's close to 100 degrees Celsius, unless it's put under an autoclave. Pasteurization is reducing the number of spoilage organisms. It does tend to kill pathogens, but it does not kill all organisms in the thing that's being pasteurized, like the milk that's being pasteurized. There are 2 ways of pasteurizing things. Mild heat, sufficient to kill organisms that cause spoilage, like pathogens. It tends to denature the proteins and enzymes, but thermoduric organisms can survive. So when you pasteurize the milk, it's not sterile. There are microbes in it. It just will last longer than unpasteurized milk, and pasteurization kills the pathogens. Any questions about that part? It does have a shelf life, and even in the refrigerator, you do need to toss it away because it is not sterile. There are microbes in it. There's 2 pasteurizations. One heating at 63 degrees Celsius for 30 minutes. Most pasteurization is heated to 72 degrees Celsius for 15 seconds. Okay? Any questions about pasteurization? Pasteurization was started by Louis Pasteur in 1857. You don't need to know the term, but you should know that Louis Pasteur invented it, and that's why it's called pasteurization. He didn't use it for milk, which is what it's mainly used for today. He invented it for beer and wine, and to tell you the truth, I don't know of any wine that's ever pasteurized, but beer is pasteurized, and I can't tell you the names of the beer which are not, but most beer is pasteurized and then bottled and then sold. And I live in Oregon, and for many years there was a health law that said that beer had to be pasteurized, and Coors beer could not be sold in Oregon because it is not pasteurized. Those of you who are Coors connoisseurs know that it's filtered, and because it's filtered, It has a slightly different taste from most other beer, what's another beer? Budweiser, Heineken, and they're pasteurized, and it gives the beer a slightly different taste. Okay? Any question about any of that? And for those of you who like cures, the Oregon legislature changed that a long time ago, something like the 1980s, and put in an exception, and that is that if the beer is filtered, it can be sold in Oregon now. So Coors beer is sold in Oregon now. There's another treatment for milk. It's not pasteurization. It's called ultra high temperature, or UHT, where the milk is brought to 140 degrees Celsius for 1 second. And then it's packaged, sealed, and this milk does not require refrigeration because there are no living organisms in them except maybe endospores. And that kills the bacteria except for maybe endospores in that milk. And so this milk can be sold on the shelf without refrigeration, and it lasts a long time, something like a year, if it's not opened. Any question about that? Anyone ever see that milk in the store? That's sort of similar to canned milk. It's not exactly ultra high temperature, but it's sterilized and then canned while it's sterile, and then because it's in the can, it can't get contaminated, and that's similar. The other method for heating is dry heat, and as you can see from this kitty who liked the oven, dry heat is not as effective to killing. Like that kitty is perfectly fine climbing into the oven. All right? Another way that you can say is when you're using wet heat and you're having steam, you cannot put your hand or arm through the wet heat. Otherwise, you'll get burned immediately, right? But you can put your hands into the oven and bring out whatever you're cooking. So you can put your hands into the oven for a short term and you will be fine. So that's demonstrating that wet heat is more effective than dry heat. Any question about that? Dry heat kills by oxidation. Flaming and incineration is extreme temperature to flame an object, but you can sterilize things by baking it by hot air sterilization, and that is like 170 degrees Celsius, 350 degrees Fahrenheit or more, and you need to sterilize it for 2 hours. So dry heat does work, but it takes a long time. And that's why it's not as effective, dry heat, and why you can put your hands into the oven and then take out what you're cooking. Short-term exposure does not burn you. Obviously, you can't put your hand in the oven for a long time, and you can't touch anything. You can touch the air. If you touch the metal, that will burn you because the transfer of heat from the metal is much quicker than the transfer of heat from the air. Any question about that? So, wet heat transfers heat much quicker, much more efficiently than dry heat. Hopefully, this kitty is demonstrating that. Any questions? And by the way, this kitty, sorry?
aren't each individually just as important? Because like you can't get gauze wet.
Well, gauze is sterilized and then put on the patient to protect the patient. But yes, if you get it wet, the microbes can come through the gauze. So gauze is meant to be kept dry. I'm saying that wet heat is more efficient at sterilizing than dry heat. And the point is that through steam, immediately you can become burned. But in dry heat, you're not immediately burned. I have no idea how long you have to have your hand in the oven before you become burned, but it's not immediate. Okay? All right. And no kitties were harmed in the taking of this picture. This kitty just liked the warmth of the oven, and the oven was cool enough before the kitty climbed in, but it was warm. Another way you can sterilize organisms is by filtration. We talked a little bit about that when we were talking about Coors beer. And the filter sterilize what's after the filter, and that is the microbes are picked through or stopped by the filter, but the liquid or the air moves through the pores in the filter. Generally, only liquids or gases can be filtered. It is useful for sterilizing liquids like Coors beer. That would be altered by heating. Some juices are filtered. Actually, some vaccines are filtered. Milk cannot usually be filtered because that would the proteins and the lipids in the milk would be caught by the filter and then would actually clog the pores, and then the filter would no longer work. Any question about that? Low temperature inhibits microbial growth. Generally speaking, with bacteria, it does not kill at least all cells. Like if you freeze something, about 50% of most bacteria would survive freezing. Refrigeration, the bacteria may just become inactive. They would not reproduce, they would not synthesize toxins, but it would slow their metabolic rate. They would not reproduce. Obviously, psychotrophs can reproduce in the refrigerator, and they can reach about 2 million cells, a psychotroph, within about a week, and then that can cause spoilage of food. And that's why when food is refrigerated, you should really only keep it for about a week in the refrigerator. And that's most food, not things containing salt like pickles or ketchup, or vinegar, which is also pickles. Like if you were to treat your onions and put them in vinegar, you could refrigerate that longer than a week. And that's because other agents besides the temperature are preventing the microbial growth. And if you don't know about ketchup, it has both acid from vinegar and saline, the salt, as well as the sugar, which are preventing the microbial growth. And that's why ketchup can last, well, over a year. I had a ketchup bottle in my mother's refrigerator for 2 years because my mother and I didn't eat ketchup. And after 2 years, I told my brother, you should take that, because he put ketchup on everything. And I said, we don't use it, and it's old, so take it. And he did, and he was fine. And the ketchup was in the refrigerator, like I said, for 2 years. And it was still good. But generally most food, like if you're making, I don't know, a cake, or, well, of course, actually that has a lot of sugar. So let's say the cake that doesn't have a lot of sugar, or most food, and you refrigerate it, you should toss it out after about a week. Freezing stops metabolic activity. It doesn't necessarily kill the cells, but you can keep food in the freezer for a year or more. Slow freezing in your home freezer, that actually does allow ice crystals to form, which can kill some bacteria cell, and about 50% of bacteria cells will die. It does not kill endospores, but it does kill all parasitic eukaryotic species. And so if you have food and there's a possibility there's a parasite in it, you can freeze it and then it'll be good. That doesn't apply to quick freezing, and that would be freezing something in liquid nitrogen, and that would not kill bacteria. But to tell you the truth, I don't know of any food that's quick frozen except maybe ice cream. So quick freezing in liquid nitrogen is not frequently done. I'm not even sure why you would do that. Does anyone know why you'd ever quick freeze something? Anyone know of another food besides ice cream that's quick frozen in liquid nitrogen? Yeah, nobody's coming up with an example. The only reason why you might do quick freezing is if you did not want ice crystals to form in the item. And I wonder if the medical industry quick freezes different things to prevent ice crystals from forming. And that would be my guess, is that in medicine they might quick freeze things in liquid nitrogen to keep the ice crystals from freezing or forming. I know you can freeze like eukaryotic cells in liquid nitrogen, and the quick freezing prevents the ice crystals from forming, which would then kill the cells. But that's to preserve the cells, not for any other reason. A high pressure can be used to kill microbial growth. This is applying the pressure evenly, usually in a liquid, and that will kill vegetative bacterial cells. It denatures proteins and alters carbohydrates. Endospores are not usually killed by high pressure. They're resistant to that. We do this high pressure killing of liquids like juice, and that's done to preserve the flavor, the color, and as well as the nutrient value. Whereas when you heat them, like boiling, and then you preserve it and can it, that changes the flavor and destroys the nutrient value. If you don't know, like if you're making homemade lemonade, if you were to boil that, you would destroy the vitamin C. But if you use a high pressure on the fruit juice, you retain the vitamin C. Any question about that? And this is applying a little joke, high pressure. Your dad is under a lot of stress lately, and he comes home and he's a diamond because they're coal. Okay? Not exactly my example, but fruit juice is frequently under high pressure. And if you look at it, a lot of the fruit juices in the store, you'll see that it has vitamin C, and that's because the high pressure doesn't destroy the vitamin C. Desiccation can also be used as a physical method to control microbial growth. That may not kill all microbes, but it prevents microbial growth. And desiccation is just drying the food out, or drying like the fruit or the vegetables, The bacteria cells can remain viable for years if there's endospores there. Resistance to desiccation varies greatly by species. Some species will die with desiccation. Like generally dried fruits and vegetables, they're not going to have E. coli on them. Any E. coli that would be on them would be something that was added recently. Like if you dry your fruit or your vegetables, they can be good for about a year, and the E. coli that was on the fruit a year ago would be all dead. Like I said, the only E. coli would be from somebody recently touching the dried fruit or vegetables. Examples of desiccation are dried fruit, like dried apricots or dried prunes. That's a plum that's been dried, or raisins. Preventing microbial growth, not necessarily killing the cells, but a lot of cells are killed by that, like E. coli. Osmotic pressure, very similar, except you have high osmotic pressure, like high salt, high sugar, and that will cause plasmolysis. The water will leave the cell, the plasma membrane will shrink and pull away from the cell wall, and that's what plasmolysis is. And that's why molasses or honey stays uninfected. The high sugar content, when bacteria fall into the cell, the water leaves the cell, and if that doesn't kill the cell, it at least stops the cell's metabolism, and the bacteria cannot reproduce in that. My mother once had a bottle of molasses that was good for 20 years, and then I made some molasses candy for my father. And he ate it, and he enjoyed it, and he survived from eating it. So that molasses stayed good for 20 years. And that bottle of molasses survived in my mother's kitchen for 10 more years because she never used it, and I only made molasses candy once. And so we figured that the molasses was good for 30 years. But after 30 years, the bottle was eventually thrown away because nobody ever used it. Okay? Pointing out that at high osmotic pressure, the organism or the molasses stayed for many, many years. All right, any question about the physical methods for controlling microbial growth? This is still a physical method for controlling microbial growth. We can use radiation as a physical method to control microbial growth. Radiation damages DNA. When we're talking about all radiation, the visible light is part of the radiation, but only a small part. Let me show this a little better. So visible light is part of the electromagnetic spectrum of radiation. When we're usually talking about radiation, we're meaning gamma rays, X-rays, and UV light. So that's lower, a longer wavelength than visible light. And we can use radiation above visible light to control microbial growth, but that's not usually what we call radiation. That would be infrared, like heat, and I've already talked about that, heat. And then above that would be microwaves, and then radio waves. Radio waves do not cannot be used to control microbial growth. Microwaves can, and what they do is they will hit an object and then heat it up, so the microwaves will be converted to heat in the object, and that can kill cells. Okay? Any question about that? So microwaves do not directly kill cells. They'll be converted into heat, and then in your potato, the heat will cook the potato, and that will cook the bacteria. And that's infrared in the electromagnetic spectrum. UV, X-rays, and gamma rays are the radiation we're talking about. And the shorter wavelength UV light cause thymine dimers in the DNA, and I'll talk a little bit more about that. And that can mutate the DNA and cause the cell to die. X-rays and gamma rays give more energy to whatever they hit, and they can ionize molecule, meaning they give enough energy to what they hit to cause the atom to lose their electrons, and then that causes ions from that molecule that lost its electrons, and the ions can then go on and do damage in the cell, ions and hydroxyl radicals, especially when they come up to DNA and then damage the DNA. So that is how radiation, at least gamma rays and X-rays, they produce ions and hydroxyl radicals, and then these are very reactive, and they can damage what other molecules they bump into. And if they bump into DNA, that will mutate the DNA, and enough of them will kill the cell. And that's how gamma rays and X-rays kill cells. Any question about that? Ionizing radiation can be used in the food industry to sterilize the food, and you can see that in like the camping food that is sterilized, or I don't know, something that's packaged to prevent microbes from getting into it. It's sterilized. Does anybody know an example of a food that's sterilized with radiation? Nobody?
Military rations.
Military rations, thank you. Pharmaceuticals can be sterilized that way, and various medical supplies. Non-ionizing radiation has less energy than ionizing radiation, and when the UV hits the DNA, it tends to cause thymine dimers, where two thymines that are next to each other, they have to be right next to each other. Instead of binding with a hydrogen bond with the A, they bind covalently with two covalent bonds between the thymines, and this disrupts the DNA. This part of the DNA cannot be transcribed or replicated, and if it is replicated, a mutation will happen because this won't be read correctly. And you get enough of these in the cell, and that will be lethal. That will cause a mutation when the DNA tries to replicate that. All right, any questions about radiation, UV or ionizing radiation? I've already talked about microwave kills by the microwave being changed into heat. So this table summarizes the different physical methods used in controlling microbial growth. Heat, and as I stated, there's two types. There's boiling or steam. Autoclaving, which is heat under pressure, and that's very effective. Pasteurization, which is not very effective, but it is a form of moist heat. And then dry heat, direct flaming, which is very effective, but that's at a high, very high temperature, and you essentially turn things into ash. Incineration, also turning things into ash, another high heat. Hot air sterilization, that's baking, and it is effective, but it takes a long time, like 2 hours at 170 degrees. Filtration, also effective. The filter stops the bacteria. Cold Not very effective at killing cells, but refrigeration prevents most microbes from growing. Not for the organisms that live at this temperature. They will grow, and that's why after a week, all foods in the refrigerator should be thrown away. But it is used to preserve foods for about a week in the refrigerator, and various drugs and cultures. Actually, if you were growing in the lab, you would put your culture, your microbial growth, in the lab, and it would last about a month in the refrigerator. Deep freezing, also used for preserving food. Ice crystals happen with that. It just preserves the food by keeping microbes from growing. And actually the microbes are not even active because they're frozen. We never talked about lyophilization. We're not going to talk about it. High pressure can be used on fruit juices, and it's used just to preserve the color, the flavor, the nutrient value of things like fruit juices. Very effective on killing everything except for endospores. Desiccation and osmotic pressure, very good at preventing cells from growing, not necessarily killing all things, like endospores will survive. Desiccation and osmotic pressure, sorry I was going all the way up there, I meant just these two, and it just preserves the food. Radiation, two types, ionizing, destructs DNA. Non-ionizing damages DNA by causing thymine dimers. This, which I didn't describe, ionizing radiation is penetrating, so you can ionize an object very nicely. Obviously it has to be contained in a container so that other microbes cannot get into it. UV radiation is not very penetrating, but it is used to sterilize the surface of things. Okay? All right, any question about any of that? If not, I think we'll stop here and we'll continue here from next time. All right? So I'll see you tomorrow.